This protocol quantifies calcium fluxes in dopaminergic neurons, which are lost in Parkinson's disease. This is useful for understanding how abnormal calcium causes dopaminergic neuron loss in Parkinson's disease. For the first time, we demonstrate spontaneous calcium fluxes in cultured primary midbrain neurons. This method can be used to dissect specific receptor contributions involved in calcium-mediated apoptosis.
Our method provides an avenue for high-content drug screens to discover specific and effective neuroprotective compounds for Parkinson's disease. These neuroprotective compounds would prevent calcium-mediated apoptosis in dopaminergic neurons.
Begin by preparing 1 milliliter of serum-free DMEM medium with 1 microliter of HSyn GCamp6F AAV per dish. Aspirate the cell culture medium from each dish and replace it with 1 milliliter of the prepared DMEM with HSyn GCamp6F. Place the dishes back into the 37 degrees Celsius incubator for 1 hour. After the incubation, aspirate the medium with AAVs and replace it with 3 milliliters of cell culture medium. Incubate the dishes at 37 degrees Celsius for 5 to 7 days, changing the medium every 2 to 3 days throughout this period of viral infection. Prepare 1 liter of the HEAPS recording buffer, 200 milliliters of 20 micromolar glutamate recording buffer, and 200 milliliters of 10 micromolar NBQX recording buffer according to manuscript directions.